Paul Cunningham is a tech CEO. He doesn't have a medical degree, let alone a background in oncology. But when doctors misdiagnosed his dog, Rosie, and her cancer advanced far beyond the reach of traditional treatments, Paul decided to take the problem into his own hands. He started feeding endless strings of questions into ChatGPT, using it as a high-powered research assistant to translate complex biomedical literature. Following the AI's guidance, Paul paid for Rosie's tumor DNA to be sequenced, and then plugged that data into Google's Alphafold model to simulate the exact protein structures driving her disease. By cross-referencing this genetic blueprint, he isolated the specific mutated genes responsible for Rosie's tumors. He then took that information and mapped out a custom mRNA vaccine tailored precisely to his dog's cancer. Now, the AI did not manufacture the vaccine. It didn't put a needle in the dog. The AI acted purely as a high-speed data navigator. Paul still had to hand that biological blueprint over to human medical experts and institutional labs to actually synthesize and administer the dose. What Paul engaged in is a very rapid technological convergence, known as SynBio AI, the merging of artificial intelligence with synthetic biology. Large language models and automated biofoundries are drastically lowering the technical barriers to advanced genomic engineering. They take the traditional design, build, test, learn cycle, which historically required years of trial and error in a wet lab, and compress it into computational seconds. This specific story is so indicative of the current landscape that OpenAI CEO Sam Altman cited the dog vaccine case as a literal, real-world example of what happens when these systems are deployed into the wild. Saving Rosie is a miraculous outcome, but it exposes a massive systemic vacuum. If a layman can map out a bespoke genetic therapy from his laptop, the democratization of biotechnology is vastly outpacing the regulatory frameworks we rely on to keep that technology safe. Fortunately, this exact same power is already being harnessed safely within the modern hospital environment. Top-tier research institutions are integrating SynBio AI directly into their human oncology pipelines. For instance, researchers at the Garvin Institute have developed a new AI tool called AANet. Its job is to process the immense, chaotic diversity of individual cancer cells operating within a single tumor. This visualization shows how AANet simplifies that cellular chaos. It organizes thousands of individual cancer cells into 5 distinct clusters, or archetypes, each with specific biological behavior. By tracking these archetypes, the visual morphs to show a clear predictive survival curve. Machine learning models use this exact data to accurately forecast patient survival outcomes for highly specific treatments, like immune checkpoint inhibitors in urothelial cancer. This workflow is broadly known as clinical decision support, or CDS. The AI functions as an ultra-fast data cruncher, mapping complex probabilities across thousands of variables, while the human doctor interprets the output. Clinical decision support is the institutional answer to the risks of DIY medicine. The AI handles the computational heavy lifting, but a human healthcare professional retains ultimate authority over the diagnosis, prioritizing patient safety over sheer speed. While a fast experimental workaround saved Rosie the dog, human medicine requires an entirely different standard of proof. Personalized cancer vaccines for humans are currently in advanced development, with massive efforts targeting melanoma and non-small cell lung cancer. Companies like Moderna are conducting rigorous Phase 2 and Phase 3 clinical trials, testing joint treatments that combine custom mRNA vaccines with existing therapies like Keytruda. The data from these collaborations with firms like Merck is highly promising, showing a 49% reduction in recurrence or death over a 5-year period. The science that Paul Cunningham accessed for his dog is the exact same science moving through oncology pipelines today. But generating a true, scalable medical treatment demands years of highly controlled, unglamorous clinical trials, rather than a single successful chat prompt. There is, however, a dark mirror to this personalized medicine. The exact same AI capabilities used to engineer a custom cancer vaccine can theoretically be used to design novel pathogens. Bad actors can prompt large language models and predictive AI to pinpoint genetic sequences that increase the virulence of existing biological agents, or even design mutations specifically meant to evade current defense measures. This creates a completely new category of threat. It is intangible, it is decentralized, and entirely digital, a problem that traditional global security measures are effectively blind to. Major treaties, like the Biological Weapons Convention, rely heavily on tracking tangible pathogens and inspecting physical laboratory materials. They are entirely unequipped to regulate the flow of computational design data. By lowering the barrier to entry for life-saving therapies, we have inadvertently lowered the barrier for biological terrorism. It turns healthcare innovation into an urgent global security dilemma. AI integration within hospitals introduces a distinct set of internal risks. Even well-funded medical systems can fail if they operate as uninterpretable black boxes, where doctors cannot verify how the AI reached its conclusions. IBM's Watson for Oncology serves as the prime cautionary tale here. The system heavily overpromised its clinical abilities, only to struggle when confronted with the messy, complex reality of day-to-day healthcare. Trained on data from specific elite institutions like Memorial Sloan Kettering, Watson's recommendations were sometimes entirely discordant when applied to diverse global demographics. We are now capable of mapping tailored cures in days rather than decades, but this capacity rests on how we manage the friction between the high-velocity pace of AI and the non-negotiable safety of human oversight.